Wilson's disease is a autosomal recessive condition and carries any symptoms. Today we're going to be talking with Dr. Jackson about Wilson's disease.
So Dr. Jackson, what is Wilson's disease?
Wilson's disease is an inherited disorder of copper handling in the liver. When too much copper is in the liver cells, it spills into the bloodstream and is deposited into other organs, mainly the brain and the eyes. The removal of excess copper relies on a particular enzyme, but this enzyme is defective in people with Wilson's disease. Copper deposits then builds up inside the body and causes damage. Wilson's disease is inherited in an autosomal recessive manner. Wilson's disease is caused by mutations in the ATP7B gene. This gene provides instructions for making a protein called copper transporting ATPase2, which plays a role in the transport of copper from the liver to the other parts of the body.
Up next, Ryan will be talking about someone who suffers from Wilson's disease, Mercedes Mendez.
Hi, I'm Ryan, and today I'm going to be talking about Mercedes Mendez. Mercedes reports that there are certain things when you have Wilson's disease that can be a struggle. Some of these things are such as eating certain foods and even finding an insurance company to help you. Some of the symptoms that she has to deal with are skin turning yellow, tendency to bruise easily, and fatigue. Although very rare, during mitosis, if the male and female both carry the Wilson's disease trait and pass on the recessive trait forward, the offspring will have Wilson's disease.
Protein synthesis begins when strands of DNA in the nucleus of cells can eventually be transferred into mRNA, which changes into tRNA. ATP7B eventually codes for making a prion protein. Now Ryan will be talking about someone else who suffers from Wilson's disease, Emily Williams.
Emily Williams suffers from Wilson's disease. She first noticed things were wrong when her legs swelled up and the doctors couldn't figure out why. Then she couldn't write, and then she was tired all the time. For the next year, she was scared for what was wrong with her. Then she was diagnosed with Wilson's disease. She became very sad and depressed. She now misses walking and talking, but continues to strive every day.
Autosomal recessive conditions are not usually present in consecutive generations, but may occur in populations with particularly high frequency of Wilson's disease.
Mutations in the gene lead to abnormal copper transporter that cannot move copper effectively or at all. The ATP7B gene is a mutation in Wilson's disease which contains the genetic information necessary to make a copper transport protein that plays a key role in moving excess copper out of the liver. Out of the 23 different human chromosomes, the gene responsible for Wilson's disease is located on the chromosome 13. Most patients have no family history of Wilson's disease. People with only one abnormal gene are called carriers. Carriers may have mild but medically insignificant abnormalities of copper metabolism. Carriers do not become ill and should not be treated. Wilson's disease patients do become ill and must receive treatment lifelong, or eventually they will develop severe lethal disease. All siblings and children of Wilson's disease patients should be tested for Wilson's disease. Other relatives who have had symptoms or laboratory tests that indicate liver or neurological disease also should be tested for Wilson's disease. More than 300 different mutations of the ATP7B gene have been identified thus far. 1 in 100 individuals in the general population carries one abnormal copy of the Wilson's disease gene. Carriers have one normal and abnormal gene. All children of those afflicted with Wilson's disease receive at least one abnormal copy of Wilson's disease gene. 1/2 of the carrier's children receive at least one abnormal copy of Wilson's disease gene.
Molecular diagnostics have improved for Wilson's disease. Screening for Wilson's disease in newborns is flexible and has been tested in limited populations, but is not yet widely performed. Identification of patients with Wilson's disease as the cause of acute liver failure is possible using standard biochemical tests. Treatments for Wilson's disease include chelating agents and zinc salts and liver transplantation. Future therapies may include hepatocyte plantation and gene therapy, both of which have been tested and shown to work in animal models of Wilson's disease. Future human studies await advances in these areas. Wilson's disease is an autosomal recessive disease due to pathogenic mutations in ATP7B. ATP7B is the only identified gene known to cause Wilson's disease and encodes a transmembrane copper transporting ATPase of the same name.